This is Holy Cross component 1 mock, and this is mock number 3. I'm on to question 4, a reasonably straightforward photosynthesis question if you have memorized your cycles. And we've got the biochemical pathways involved in photosynthesis, and it's just the stuff we expect to see. We've got, what have we got here? We've got our starting point of water, we've got our photosystems, got cyclic, and we've got non-cyclic, chemiosmosis over there, and then we've got something going around in a cycle to do with photosynthesis. I wonder what that could be. We've got, this is our photosynthesis equation. We've got water and carbon dioxide going in. We've got oxygen coming out, and our final end product of photosynthesis coming out there. Okay, right, well it all looks normal from here. Where in the chloroplast does a light-dependent reaction happen? So we should be fine with this. Thylakoid membrane. So these need membranes for all of these proteins that are involved with this process. And name the light harvesting units 1 and 2. There's a clue there, 1 and 2. These are your photosystems. And you might have actually said, you know, P680 or P700. You can't call them photosystem 1 and 2 because it's you don't get anything for just 1 and 2. What name is given to the process by which ATP is produced in the light-dependent stage? That is photophosphorylation. So phosphorylation is adding the phosphate. Photo using light. Photophosphorylation. During the light-dependent stage, so this stage here, water molecules are broken down to produce oxygen, electrons and protons with reference to the diagram. So all of these in part D need to be not just made up by you, they need to be talking about the diagram. Name the process by which water molecules are broken down. So water here, broken down, you should be fine with that. Broken down, it means lysing, broken down by light, photolysing, so it's photolysis. What happens to the electrons? Well, here's the electrons. Let's just show you where our electrons are in this, and we'll also show you where our protons are in this. There we go. So what happens to the electrons? Well, you can see the electrons going to the light harvesting units, the photosystems, but we need to be a bit better than that. We see electrons being lost from this system in the non-cyclic, and so these electrons from water are replacing the lost electron from photosystem 2. So replacing electrons lost from photosystem 2. My sympathy if you said they're going to get raised to a higher energy level and so on, but that's not really what the question was wanting there. What happens to the protons produced in this process? Well, there's our proton, our hydrogen ion. As we can see, it's going to be joined onto NADP to make NADPH2 in this diagram. So it's accepted by NADP. Or it reduces NADP. There's another way of saying that. What is the function of A? What is A? Can't find A. Ah, there's A. So we can see this is electrons are going towards it and moving away from it. So the right words to use there are accepting electrons or donating electrons. So accepting or donating electrons. Those are our cytochromes or electron carriers, electron acceptors. In the light independent stage, so it even tells you here what we're talking about. Name the pathway. Name the pathway. Okay, you should be that. The pathway that in photosynthesis that goes round in a circle can only be one, which is the Calvin cycle, or Calvin-Benson cycle. Where in the chloroplast do these reactions take place? So in a different place to the one before, and you should be happy that these take place in the stroma. So we can draw a little diagram there. Here's a chloroplast with a double membrane. Here's thylakoids and the stroma is the other stuff. In the chain of chemical reactions, which of letters A to D indicate a 5-carbon compound? Well, our CO2 is a 1-carbon. That's joined onto our 5-carbon, and then that would make a 6, which splits up into 2 3s. So these are 3 carbons and 3 carbons. And going back to our 5-carbon, that's where we're losing carbon for the products of photosynthesis. So our 5-carbon compound is C, which would be RUBP, ribulose bisphosphate. But we don't need to remember that, we can just put C down. Mark with an X one other place where ATP is broken down. Well, ATP is also it's used. The one it's not talking about is used down here in the cycle, building TP, but also regenerating our carbon acceptor. So regenerating RUBP. So we're going to put, what do we need to put? Mark with an X. So there's our X there. So ATP is used in regenerating our RUBP. Fill in box 1 to indicate the end product of photosynthesis. And just go back to your original equation, photosynthesis equation. What gets made? So we're going to have a glucose there. And that should have been a fairly straightforward 12 marks, hopefully.